Yeah, I'll continue from the point where I had left in the previous video. So we completed up to here, right? The function of nicotinic NN receptor is depolarization in catecholamine release also. Now, what is the transducer mechanism? Since these nicotinic receptors are kind of ligand-gated cation channels, these channels, when receive the acetylcholine, these channels open a way for the positive cations, okay, positive ions. And when the positive ions get inside the cell, this electropositivity inside the cell increases. And when the electropositivity reaches the limit to produce threshold potential, then that induces the axon potential, which travels along the nerve fibers in the form of nerve impulse. Okay, so the transducer mechanism is just opening the cation channels, that is sodium and potassium cation channels, and for the NN nicotinic receptors also the transducer mechanism is the same. When the acetylcholine binds with the receptor, then it opens its way for the sodium, even calcium and potassium ions. And what are the agonists? Agonist of the NM receptor. That is phenyl trimethyl ammonium. Okay, you can remember this chemical structure for you to make it easy to remember. Phenyl trimethyl ammonium, and the agonist for NN receptor can be dimethyl phenyl piperaginium. Okay, dimethyl phenyl piperaginium. Okay, there are many examples actually, but I've just written here the important examples from exam point of view. And these chemical structures are not important from exam point of view too, but it'll be easy to remember. Okay, and if it's difficult for you to remember, you can make the chemical structure of your own too. Okay, for example, this is not correct chemical structure, right? But you can use it to make you easy to remember. For example, this describes dimethyl phenyl piperaginium. Although this is not correct, you can use it for yourself to remember, but never ever write this structure in exam. Okay, because correct structures are this one. Now what are the antagonists? What are the antagonists of the NM and NN receptors? So out of many, I'll write the important one. The antagonist for the NM is tuberculin. This is the chemical substance, and in the olden days, our ancestors used to put the tuberculin on the tip of the arrow, on the tip of the arrow, like this, and they used to put the tuberculin here, and they used to shoot the arrow to hunt the animals, and that would paralyze the animals, right? And the antagonist of NN receptor is hexamethonium. Hexamethonium. I only use now the pen. Hexamethonium. Okay, so tuberculin is the antagonist of the NM, and hexamethonium is the antagonist of the NN receptor. Now that we have finished the remaining part of the previous lecture, now we'll get directly into the adrenergic system. First, let me align this paper correctly. Okay, yes. Now we're starting adrenergic system first. So what is adrenergic system? Autonomic nervous system has two parts, that is parasympathetic nervous system and sympathetic nervous system. Okay, and since parasympathetic nervous system uses acetylcholine as the sole neurotransmitter, it is also called cholinergic system. And since sympathetic nervous system uses a different one, that is adrenaline or even noradrenaline, it is called adrenergic system. Okay, and there's one thing. Suppose this is the preganglionic fibers, right? And the preganglionic fiber of adrenergic system uses acetylcholine, okay? But the postganglionic fiber uses adrenaline or noradrenaline. But in case of parasympathetic fibers, both pre and postganglionic fibers, they use the acetylcholine as their sole neurotransmitter. Okay? Parasympathetic fibers uses acetylcholine in both pre and postganglionic fibers, but the sympathetic or the adrenergic system or fibers uses acetylcholine in the preganglionic fibers and adrenaline or noradrenaline in the postganglionic fibers. So since adrenaline has been used, this system is also called or named as adrenergic system. Yes, now we're going to talk about the types of the adrenoceptors. Okay, types of the adrenoceptors. So basically there are two types of adrenoceptors, that is alpha receptors and beta receptors. Okay, alpha receptors and beta receptors. And alpha, there are basically two types of alpha receptors, alpha 1 and alpha 2, and in beta there are basically there are three types, beta 1, beta 2 and beta 3. Now we are going to talk a little bit details about this alpha 1, alpha 2, beta 1, beta 2 and beta 3. So let me turn the page here. Yes, now we will talk about the characteristics. Okay, characteristics of the adrenoceptors. So under characteristics, so let me describe the location. Location of alpha 1. This alpha 1 is present in the post junctions of the effector organs. Suppose this is your muscle and it is commonly found in blood vessels. So let me draw blood vessel. Suppose this is your blood vessel, okay, and this is your alpha 1 receptor. Okay, this is your alpha 1 receptor, and this is the post-junctional effector organ, and the receptor present in the post-junctional effector organ, this alpha 1, it receives the nerve impulse from the neurons in the form of the neurotransmitter. Okay, so basically what I wanted to tell you is this alpha 1 is present in post-junctional location on the effector organ. And what is the location of alpha 2? Alpha 2 is present pre-junctionally, okay. If this is junction and this is in junction, okay, this is synaptic cleft, and this is preganglionic fiber, this is postganglionic fiber. And post-junctional means alpha 1 is present post this junction, but this alpha 2 is present here, even in the nerves, okay, pre-junctionally in the nerves. And if this secretes ACH, this ACH again comes here to inhibit its own production. Okay, so this is prejunctionally present in the nerve endings, and it is also present in the blood vessels, even in pancreatic cells, especially in the beta cells, and it is also present in platelets. Okay, and what are the functions then? So functions, I will describe the functions or actions in details in another lecture, but here I'm going to talk a little bit about this. So the functions can be, in liver, glycolysis occurs during this. In the heart, it causes arrhythmia, and in genitourinary system, it causes smooth muscle contraction, and in glands it causes secretion. In gut, it causes relaxation of the smooth muscles. Okay, and in blood vessels it causes vasoconstrictions. Okay, and the functions of alpha 2 can be, prejunctionally in the nerve endings. It inhibits its own production. Okay, inhibition of the neurotransmitter release, and in blood vessels, vasoconstriction, and in pancreatic beta cells, it helps in decreased insulin release, and in platelet, it helps in platelet aggregation. Okay, so transducer mechanism. What is the transducer mechanism? Transducer mechanism is similar to the M1 and M2 receptors. Okay, that is the same IP3 and DAG pathway, and this IP3 DAG pathway here, in this pathway, the IP3 causes increased release of the cytosolic calcium, and that causes increase in the phospholipase A2 concentration, and that increases the PG release, and these all the cascades causes electropositivity of a cell, and hence activates or excites the cell. And here, since this is inhibitory receptor, the transducer mechanism of alpha 2 and M2 receptor are similar. That is, decreased CAMP production and decreased calcium production, or decreased calcium influx, and then increased potassium efflux. This all causes the cell to be more electronegative, and hence the cell is deactivated. And what are the selective agonists? Selective agonists, namely a phenylephrine, and another what is that? I forgot. A methoxamine, and another selective agonist for alpha 2 can be clonidine. Okay, and the selective antagonist for alpha 1 can be prazosin, and terazosin. Okay, and these are drugs that are used in benign prostate hyperplasia. You will know it later. And the selective antagonist for alpha 2 is yohimbine. Okay, yohimbine. Now we have talked something about alpha 1 and alpha 2. Now let's get into the alpha beta receptors. Okay, we need another page. So beta receptors. Beta receptors, there are 3 beta receptors: beta 1, beta 2, and beta 3. Okay, and location of the beta 1 is, it is located in heart and Jg cells, and these are located in, I will give mnemonics. Just remember the mnemonics: BUBLE, bubble gum. BUBB, sorry, yeah, BUBBLE, bubble, GUM, bubble gum. But from this bubble gum, remove this B and remove this M. So there remains BUBLEGU. So this B stands for blood vessels. This U stands for uterus. This B stands for bronchi. This L stands for liver. This E stands for eye. This Z stands for gastrointestinal tract, and U again stands for urinary tract. Okay, and beta 3, this is very important from exam point of view. Beta 3 is present in adipose tissues. Okay, so what can be the functions? So beta 1, these are stimulatory, and beta 2, beta 3, these all are stimulatory. The receptors that we have talked so far, among them only alpha 2 and M2 are inhibitory. Do you get me? Every other receptors are excitatory. So since beta 1 is also excitatory, it increases the heart rate, it increases the contractility, it increases the conduction, and it promotes renin release. Okay, renin release, and in blood vessel it causes vasodilatation, the beta 2 receptors, and in uterus it causes relaxation, and in bronchi it causes bronchodilation, and in liver it causes glycogenolysis and gluconeogenesis. Okay, and in the eye it causes dilatation of the pupil or mydriasis, and in GIT what it does is motility of the GI decreases. Motility of the GI decreases. Okay, and beta 3 receptor does lipolysis. Okay, since this is present in the adipose tissue, it causes lipolysis. Okay, now what is the function of beta 2 in urinary tract? It causes in urinary tract, it causes increased tone of the the sphincters, increased tone of the sphincters. Okay, now what is the transducer mechanism? The transducer mechanism is as I already told in the previous lecture. Where is that paper? I like to search that again. Okay, leave it. I'll make it again. Suppose this is your cell, and this is your receptor, and when the neurotransmitter comes, the G protein, which have alpha, beta and gamma component, this breaks into alpha, beta, and gamma separate entities. It occurs in expense of GTP, right? GTP changes into GTP, and the energy released from here breaks this protein into alpha, beta, and gamma. And this alpha, what it does is this inactivates the adenyl cyclase. And when adenyl cyclase is inactivated, okay, this alpha, beta, and gamma, these are separated by the energy released from the conversion of GTP into GDP, and this alpha, this goes and inactivates the adenyl cyclase enzyme, and this adenyl cyclase, which facilitates the conversion of ATP into CAMP, okay, and this process is decreased, or this process goes down, and when CAMP goes down, calcium level inside the cell also goes down. This was actually the mechanism or transducer mechanism in case of M2 receptors. But here, since this is excitatory, what happens is this adenyl cyclase is activated. Okay, and when this adenyl cyclase is activated, this ATP conversion into CAMP is increased, not decreased, right? And when CAMP level decreases, calcium level also goes up inside the cell, and hence the cell becomes more electropositive and is excited. So the transducer mechanism here is increased CAMP, increased calcium influx, okay, and here also increased CAMP, increased calcium influx, okay, and here also the same mechanism, beta 3, increased CAMP and increased calcium influx, okay. And let me write here, yeah, what are the selective agonist for beta 1, yeah, Dobutamine is the best example, and beta 2, you might be hearing this in treatment of asthma. That is, selective agonist for beta 2 is salbutamol, okay, and selective agonist for beta 3, the name is but difficult to remember, so, and it's not even important from exam point of view, so I don't want to discuss here. And selective antagonist for beta 1 is metoprolol, and for beta 2 is alpha methyl propanolol, okay. So we have now discussed about the beta receptors, right, in a few words.